This is paper 1, question 3. I love this question. You've got to love a question that's got a picture of a bee on it. So this is pollination in insect pollinated flower. So we've got a bee. It's quite a nice bee. This little tongue going down there. So I'm thinking that R must be a nectary at the bottom. Then we've got where to start really. There's the stigma, style, ovary, stigma, style, ovary. So these are that's an anther. And this one shows the anther hitting the bee there and depositing pollen. And this one shows the anthers have shriveled, died, dead, done their job. And this one has got the ovary style, and that's got the stigma hitting the bee, so this is picking up pollen. And we've got a dead stamen. Oh, it's labelled as a dead stamen, and the bee's tongue. How nice. So let's have a look at the questions then. So I'm already thinking if these are died before the stigma picks up pollen, I'm already thinking, well, that's a great mechanism for ensuring cross-pollination. So main parts P and Q. P is the stigma, and Q is the ovary. Name the substance produced by structure R. Well, that's where the bee's proboscis is going, so then it'll be slurping up nectar. And what's the function of this? Well, nectar attracts insects. So handy to have if you're insect pollinated. Describe what happens to the pollen in diagram 1 and 2. So in diagram 1 we've got the anther, oh, you can't see this, anther poking at the bee's back. So the pollen, describe what's happened to the pollen. Pollen is deposited onto the bee. Assuming it's a bee, it looks stripy. And in diagram 2, where the stigma is touching the same bit, the pollen is transferred to the stigma from the insect. I'm going to call it an insect now because I'm not sure it is a bee. So, using the diagrams opposite, explain how these flowers are adapted to ensure that there's effective pollen transfer. So here we've got the idea that the pollen is deposited at the same part of the bee that the stigma touches. There'd be no use depositing the pollen on the legs and trying to pick it up from the back. How do we avoid self-pollination? So this is our idea of our dead stamen in diagram 2. So the anthers mature before the stigma does. Some of you may remember that term, protandry, which just means male parts develop before the female parts. That's all looking good. Oh, pollen grains. Pollen grains, always interesting. So we've got our megaspore, we've got division, We've got our microspores, and then we've got the mature one with its exine and 2 nuclei. So, questions. Which floral part does this take place? Anther. Name the cell process represented by X and Y. So this is going from 1 cell to 2 cells to 4 cells. You need to be careful and make it really clear that you can spell meiosis. One of the few terms that you have to have the correct spelling. So I remember it, meiosis. Diploid number is 10. Underneath each structure indicated, write the number of chromosomes in each nucleus. So we start off diploid, split the homologous pairs, which takes it down to 5, and then divide the chromatids. 10, 5, 5. Give the functions of the generative nucleus. So this is the one that divides into 2 and does that double fertilisation trick. So divides into 2 by mitosis. 1 nucleus fuses, so I don't want to say fertilises, rubbish. 1 nucleus fuses with the egg cell to form a zygote. The other is the polar nuclei to form the endosperm. Now I'm thinking that's 1 mark. They probably don't need all of that, but why not let them know that you know this stuff? And you don't know what they're going to allocate that mark for. And the tube nucleus, lastly, this controls the growth of the pollen tube. And enzyme synthesis to digest the pathway through the style. Again, possibly a bit much for 1 mark, but you don't know what that's going to be allocated for. You don't know whether it's for enzyme synthesis or whether they just want controls the growth of the pollen tube. Give them an all. Don't stint.